Welcome to Ask the Expert. We've got Dr. Mark Huising here. He leads an independent research program at UC Davis that is focused on the physiological role of paracrine crosstalk within pancreatic islets. And this work really builds on a novel negative feedback loop that his lab actually discovered, where the peptide hormone urcortin, that's a mouthful, 3, released by beta cells in response to glucose, is necessary to promote somatostatin secretion from the pancreatic delta cells. Delta cells don't get a lot of, love or press. So this is going to be very interesting to see what's going on here. So somatostatin in turn inhibits the beta cells to prevent excellent insulin release. And by visualizing the behavior of multiple islet cell types simultaneously and in real time, the Huising group quantifies and compares the functional responses between the main endocrine islet cells types in response to hormone, nutrient, and neurotransmitter stimulation. So this comparison of functional responses across populations of hundreds or even thousands of individual cells really reveals a significant functional heterogeneity among alpha, beta, delta cells that really extends and goes far beyond the subset of immature virgin beta cells described by his group a few years ago. So we cannot wait to see what you are going to share today, and welcome. Thanks so much for joining us. Ooh, this looks good.
All right. Thank you, Monica, for the introduction and for the opportunity to talk about the work that we do. And unlike conventional talks where I just try and set up and tell a complete scientific story, what I figured I'd do for this one is to focus really on show a couple of vignettes with hopefully pretty videos, and then maybe spend a little bit of time on the methods that we use, which are how we are able to image. They're not the only ones, the only methods of doing that, but maybe as a point of starting point for a discussion and the Q&A as to how one goes about visualizing behavior of islet cells in real time. So as we all know, everybody in our field, for all the right reasons, focuses on beta cells because the beta cell in the islet is the only cell that, when dysfunctional, causes disease. Type 1 diabetes, when we lose our beta cells in an autoimmune mediated attack. Type 2, when beta cells fail over time because they're asked to work too hard in the face of peripheral insulin resistance. And more rarely, but no less impactful, diseases of hyperinsulinemia, where excess insulin can cause dangerously low or even fatal hypoglycemia because the beta cells are unable to cease releasing insulin in time. Testament to the fact that insulin is a really powerful hormone that needs to be managed carefully, as anybody with type 1 diabetes will tell you. So the focus of my lab, and I'll set this up in this slide, is that the islet contains more than beta cells, even though I just said that the beta cell is bar none the most important cell in the islet. And there's also alpha cells and delta cells, and they communicate. There's no coincidence that they occur in the same islet together. There's extensive crosstalk going on, and we're starting to unravel that. Principally, there's glucagon, which traditionally we position as a counter-regulatory hormone, really important in preventing dangerously low hypoglycemia. However, I think more recently, I think there's been a revisiting of this old notion that glucagon is actually able to stimulate beta cells under high glucose, which mechanistically is not surprising at all. And we're now revisiting the potential physiological relevance of that. But the view actually starts to shift from during the prandial phase, when we digest a meal and take up nutrients, where glucagon actually becomes cooperative with insulin in pushing additional insulin release rather than being its functional antagonist, as the textbooks traditionally have you believe. And then finally, we have delta cells, which make SST, somatostatin. Somatostatin is a quintessential inhibitor. Wherever you find it, it will inhibit. And in the islets, it will inhibit beta cells and alpha cells. We've known this for a long time. The physiological importance of this is often overlooked or even understudied. And I'm not going to go into much detail, but there is a direct physiological role of delta cells, and one can quantify that. Now, the overall image then, or picture then, is that what we tend to call glucose-stimulated insulin secretion. Now, you take some islets, whether it's mouse or human, you put them in a dish, you add glucose, and you measure insulin release, and we say, okay, this is the insulin that's coming out of the islet in response to glucose stimulation. what actually is going on there is it's the direct beta cell autonomous response to nutrient stimulation, potentiated by an alpha cell dependent mechanism and restrained at the same time by a delta cell mediated feedback inhibition. And then just to illustrate how incredibly complex this can be, just in the islet alone, there are hundreds. This is just the top 150 most abundant GPCRs only. This is not even talking about any other receptor or channel expressed. Some of them in all islet cell types, some of them selectively in alpha, beta, or delta cells, as this schematic kind of tries to convey. And all of these do something. All of these mediate an activation or an inhibition of one or more islet cell types. So, and we understand some of these, and others we are just now starting to understand. Okay. So the next slide, I'm going to try and illustrate the complexity of islet behavior. And that slide has sound. So you may want to keep your finger at the volume button in case it is a little bit too loud on your end. I think it should be okay, but just as a heads up. So what you're looking at here is an islet that is responding. The readout here is calcium. This is an islet where we use a genetically encoded calcium sensor, GCAM6. And the cells you're seeing firing here on the basal fasting glucose are delta cells. And they're always active. There's always this basal activity going on that we think is important in conveying conveying delta cell function. And then as we shift from low glucose to high glucose, we then start to see beta cells chime in and become activated. And then if we add amino acids, alpha cells also start to chime in. So now we actually see what's going on in response to a mixed meal stimulation or an epinephrine stimulation at all cell types in the islet at the same time. And it just hopefully conveys a little bit of the complexity of those behaviors that are going on all at the same time.
This is just beautiful. It's amazing.
So this is a trace that's probably an hour and a half long. And we're just imaging this near continuously. There's like probably a 5 to 10 second frame rate here. And then we purposefully image all these cell types at the same time. And then what we do is we fix these islets and we stain them. And then you can come back and find the exact same islet. And by conventional whole mount staining, confirm that the cells that behave like alpha cells indeed express glucagon, the cells that behave like delta cells and indeed express SST. So we have a really high confidence that the cells that we record from are also the cells that we think that they are. And then you can do neat tricks. I'm not going to set up the whole story here, but you can do neat tricks like, okay, ask the question, these delta cells are constantly active. What happens if we remove them? There's ways to remove them. This is work by a grad student of mine, Jessica Wang, who has done phenomenal work setting up this model where we basically are able to in vivo ablate delta cells and then later on take out the islets. And as you can show, that model works. What it does is it causes a shift in the set point for glucose. This is not a surprise because SST inhibits insulin. If you remove the inhibition, you get a little bit more insulin coming out, leading to a reduction in the set point for glucose. So this green line of islets with no delta cells, these mice have a stable, lower glucose set point than control islets, control mice that have the same transients but did not receive diphtheria toxin. And there's a lot more data behind this, but we can also then use imaging as a readout to kind of visualize what happens here. Because what happens here, if we now, long story short, create a quadruple transgenic mouse, we first ablate delta cells, then express GCAM6 in only beta cells, and then expose islets with and without delta cells to a glucose ramp, and then test how long it takes. At what glucose concentration do beta cells start to respond to glucose as a measure for glucose threshold? And if you do that, and again, I put sound underneath here so you can differentiate also audibly what happens in the absence and the presence of delta cells. So what happens in this scenario is simply, and hopefully you can see as well as hear it, you see beta cells in the presence of in the absence of delta cells respond by 1 to 1.5 millimolar of glucose earlier, which is exactly the change in set point that we observed in vivo. And then again, we can do we can take a still of the live video and then do a post hoc video and confirm that delta cells indeed were ablated on the right and we can track every single cell we recorded from. Now, what we also can do is start to look at coordination of different cell types. I'm going to show you a video here that shows delta cells kind of firing in a random pattern, but then as beta cells come on, you see delta cells becoming entrained by those beta cells. So delta cells on their own are firing, and as soon as beta cells come on, delta cells also shift on and stay on. And these two different behaviors are interesting. And it raises the question, what is the mechanism behind that? Again, we can post or control this. And it's just visualized here. This is work by another grad student, an MD PhD student, Mohammed, who is showing that, yeah, you can get these very fast calcium spikes in these delta cells, but in response to sustained high glucose, a good number of these delta cells become entrained by neighboring beta cells and show that oscillatory pattern that is slow, superimposed on which you have these fast spikes. And then he does something like, so he's fax purifying delta cells away to demonstrate that if we remove delta cells from beta cells, that slow entrainment that is present here goes away, demonstrating that delta cells are entrained by beta cells because this behavior is not obvious, which sets up two different pathways. Either there's paracrine crosstalk or there's gap junction connections. And we're pursuing exactly what the mechanism is. We have a pretty good idea that it's probably predominantly paracrine and not fully paracrine. And just to illustrate how one goes about doing this without going over the whole data set, for which there's no time here, this is basically a set of islets where we are simultaneously visualizing both beta and delta cells. And I'm going to simply take you through it. It's an 80-minute recording. And we're going to go through a couple of different stimulations. Tolbutamide will activate both delta and beta cells, and then we're going to wash that out. And then we see delta cells retaining activity. And now we're going to go with glucose, and you'll hear delta cells come on first, and then beta cells. Then we're going to add diazoxide, which we know will inhibit beta cells, and because it opens KATP channels, it'll do the same thing to delta cells right here. So that's what silencing sounds like. And then we wash the diazoxide out, and we'll see both of these cell types come on again. And then what you do is you come back with an L-type calcium channel blocker, isradipine, which will also silence all beta cells and most delta cells. There we go. So now they're silenced again. A few delta cells remain active, and that's probably reminiscent of an R-type calcium channel mechanism that also has been reported for delta cells. So the point of this then is to show that both that basal spiky behavior that I showed earlier that is present in these delta cells is fully gone if you prevent depolarization, which I think tells us a lot about whether or not gap junctions are involved in that mechanism. And that's the story we're pursuing. It's a part of a larger story that we're pursuing. The approach-wise, though, what this does, just a single trace gives us in-depth temporal resolution on 79 delta cells and 122 beta cells in just a couple of hours. So the amount of throughput and the resolution of the data that we get, both spatially and temporally, is really quite helpful here. Finally, last really brief thing yet is work by now Dr. Glenn Noguchi. He just graduated last week, who has done wonderful work on a story where he's been able to visualize the behavior of alpha cells in response to a number of textbook activators of their signaling and secretion. And what you're going to see on the left is calcium, on the right cyclic AMP in response to amino acids, in response to epinephrine, and in response to the posterior pituitary hormone AVP, which are all known to activate glucagon secretion. But their effect on the behavior of calcium and cyclic AMP is different. That's summarized here. Calcium is activated in response to all three of them, cyclic AMP only in response to epinephrine. But what also is interesting, and what I want to draw your attention to here, is that not every alpha cell responds to all these stimuli. All of them respond to AVP, but there is a subset of alpha cells that doesn't respond to epinephrine, and a different subset that has no response to amino acids. And what Glenn then did, among many other things that I'm not going to go into, is he basically did a very similar trace, back-to-back the same protocol, to demonstrate that this is not some kind of random stochastic event, but if you stimulate the same set of alpha cells twice with the same protocol, 3 hours apart, the same cells that did not respond the first time around to, say, amino acids or epinephrine continue to not respond a few hours later, meaning that there is at least some stability to the heterogeneity, and it's probably underlying some biology. So then this is the actual setup that we use, one of two microscopes that we have, where it's a resonant confocal microscope, where we have a perfusion system hooked up that basically runs the whole thing on its own. So we program the microscope, we program the perfusion, and we're able to walk away and come back when the trace is done. And that's really helpful, because if we run traces that are like 3-4 hours, we can actually just run them overnight, come back the next day, and collect the data. This is the actual video that I took, that you just saw getting taken on the microscope, now played back for you. The biology of this is not necessarily all that important, but it is nice to be able to just measure across, in this one video, hundreds of beta cells at the same time. So how do we go about doing this? We use a suite of Cree drivers. For beta cells, we use predominantly urocortin-3 Cree. It's a phenomenal beta cell specific Cree that is really specific and hits every single beta cell, almost every single beta cell. We also use the INS1 Cree ER if you have to have some tamoxifen inducible. For alpha cells, we're using Klaus Kessler's Octagon Cree ERT model. And for delta cells, we use a constitutive SST Cree that has the additional benefit of being a functional null. So if you breed it to homozygosity, you actually get a knockout mouse for SST, which is also very helpful. And then we've done most of the imaging that I've shown you today with the GChem6 calcium sensor and the Kemper sensor. And as I said, we use a resonance scanning confocal microscope with 4 or 6 lasers, depending on what reporters we use. We perfuse with a pressure-based system. Essentially, it works just like a flow cytometer, where you pressurize the contents of your tube that then get pushed into some microfluidic tubing and gets flown over your a little valve that you can control to basically switch between different channels. We have up to 11 channels on the system. It's a commercial system from a European company called L-Flow, and it's been really helpful to us. We adapted it on in a few ways, but it pretty much is out of the box with a few minor modifications. And then we make our own imaging chambers. And there's basically a mold that we design and 3D print in-house, and then we plasma bond that to these imaging dishes. What's on the figure here is an outdated version. We basically just made a straight slot. That's what we use these days, and we can perfuse for hours on end with 200 microliters a minute. Islets are perfectly fine; they don't move, and they are behaving beautifully for us in the microscope. And that too is just really nicely stable, so we don't have to worry about like spikes in flow messing up the stability of our imaging. And that's so these molds, you basically just print these, you make 9 of these chambers at a time, and you add a little in lead and an out lead that you punch in there, so you can then slide some microfluidic tubing in there and connect this to the perfusion system, and then off you go. So that's how that works. For us, it's been working really well. I'd be happy to share details if people are interested in knowing more, but this is the gist of it. And I think with that, I'm going to end on this video. Some of you might have seen this. It's one that we took serendipitously. A student of mine, Sharon Lee, came to find me saying, "Look what I'm finding." She basically found this macrophage, or we think it's a macrophage, that is kind of crawling through the islet and kind of extending its pseudopodia, behaving like a macrophage does. And it just happened to hang out in the focal plane long enough for her to get this absolutely beautiful video in the midst of beta cells behaving. And with that, there are a lot of undergrads who contributed to this, and a really talented group of grad students. I need to give a shout out to Marcus Flischer, my lab manager, who's done a phenomenal work in setting up a bunch of this up, along with Glenn and Jessica and Mohammed, whose work I shared, and Sharon. And last but certainly not least, Alex, who is my bioinformatician. He also just graduated, but he also is the person writing all these codes that we use to quickly extract and summarize, both visually and audibly, the traces that we collect. And with that, I will shut up and hopefully be able to field some questions from the audience.
Wow, that was really elegant and so so interesting. The whole setup that you guys devised, and then the results you got from it are just really extraordinary. I see there's a bunch of questions. Let's see, is anyone interested in asking a question? You can raise your hand, or you can I think you can unmute yourself. If you can't, I can. Let's see. This is, I think, from a lay person. Tamoxifen is used in cell biology research. It's a chemotherapy. chemotherapeutic agent. Yes, that is true. I had a question, you know, I mean, we're hosting Richard Benninger in January, and his talk is going to be really based on, you know, is there a pacemaker in the islet or not? And so, and sort of in talking with people like Jacob Heckscher Sorensen, who's at Gubra, he says you know, he's very into the visualization of the islets. He says he sees that. He doesn't have this sort of level of, you know, kind of monitoring that you guys have, but he's really just interested in the optics, and he's sort of seeing something like a pacemaker. In his conversation with Decio Zurich, he feels that there's likely one too. And
well, and we have Guy Rara here
thoughts?
who also, I think, can speak to that with a lot more authority than I have. I've been following that field for with a lot of interest. And it's clear that there is always a cell that responds first. And I think between Guy and Richard, I think we have a few of the folks who have really led that, along with Dave Hodson, of course, who did that work with Guy together. Do you want to speak to this guy?
Yeah, I'd love to hear from him.
Sure, thanks. I mean, yeah, there is some skepticism in the field around whether these exist, but I think the experiment that we did with David in 2016, showing if we clamp what we've identified as a highly connected cell using an optogene, we suppress connectivity and calcium dynamics across the planar imaging, says that that cell, if it's not a pacemaker, it is at least required to transmit calcium waves. So proving that something is actually, you know, the originator and is driving in the way that you'd see cells in the AV node driving cardiac contraction is more difficult to prove. I mean, the refinements that we've made in subsequent years indicate that there are multiple sets of these, or multiple subsets of beta cells, some of which are wave originators, some of which are leaders, some of which are very highly connected. And Richard Benninger, who you hear from, has much of the same data. So it's more complex, I think, than we proposed 5 years ago. And we now have to understand this hierarchy of these subsets of cells. But as I said, identifying a bona fide pacemaker is not straightforward.
Yeah, that's well said.
And I'll just add to that real quick, which I think is an outstanding question that basically literally goes beyond the pacemaker in the single islet, and that's how the whole thing is connected in situ, right? Because we also know that insulin output is synchronous across the whole pancreas, which clearly must involve some neural mechanism, and how then that is basically imposed upon and perhaps even supersedes the islet autonomy, if you will, to respond at will. Because for that to happen, you have to have all the islets or most of the islets in a pancreas responding synchronously, and whether they do so by innovating these a subset of cells that then basically give out marching orders to each individual islet that make all islets respond at the same time. I think that's still unresolved, and it would be very nice to be able to visualize that. Clearly, our system is incapable of doing that kind of question.
Yeah, I mean, that kind of mesoscale technology you need to analyze at scale across, you know, many millimeters with cellular resolution. It sounds as though it should be easy, but it's extremely difficult to achieve technically.
What about the overlay of the sympathetic nervous system? I would be very interested to hear your commentary on it and its impact on control.
So I don't know if sympathetic nervous system, I don't think that sympathetic nerves are controlling the pacemaker activity of the beta cells. I think there is an important pacemaker, there's the potential for sympathetic input on the alpha cell. Alpha cells are exquisitely sensitive to epinephrine, although the physiological contribution of that, I think, is not, I mean, it's debated. I know people dismiss epinephrine as just not relevant for activating alpha cells, and nevertheless, it's arguably the most potent agent that we have in doing so, at least under the microscope. And teleologically speaking, there probably is the capability of alpha cells to respond so robustly for a reason. So I subscribe to the notion that if there is something there that is so robustly eliciting a response, it is not just sitting there idly for no physiological reason. So there likely is a scenario where, whether it's during a fight or flight response, where your epinephrine kind of comes in and is able to simultaneously in one fell swoop silence the beta cell and activate alpha cells, and kind of make sure that you preserve nutrients when you need them. Whether epinephrine and sympathetic activation is important for counter-regulation on the other side of the equation, I think you'll find differing opinions on that, and I think that's still I think there's still opportunity there to revisit that and also compare that to some of the other molecules that have more recently been revisited as potential activators of alpha cells under those circumstances, particularly AVP in the work by Linford Bryant.
Yeah, we've been talking quite a bit recently to Zachary Freiberg. He's at the University of Pittsburgh, and he's really interested in kind of qualifying and quantitating the neurotransmitter sort of milieu that interfaces with the islet cells, and he's really, he approaches it from a totally different way, right? He's an MD PhD, but he's like a clinical psychologist. Psychiatrist.
Yeah,
So
I'm familiar with his work. It's fascinating. I mean, there is so much,
cool.
There is so much potential. I mean, there's so many different receptors there. I mean, the potential for, there's so many different neurotransmitters and peptides and nutrients that are able and capable of activating or sometimes inhibiting the islets, that the benefit of being able to visualize the system in its completeness, even if it's only one second messenger at a time, it kind of scratches the surface as to how complicated the islet actually is. And here we are looking at a system that is just 3 endocrine cells, and that is already so incredibly complex. But I do think there is value in visualizing the whole thing, not just under one condition and at one cell type, but at least multiple cell types at the same time. Ava's asking a question about epinephrine on delta cells, and delta cells are much like beta cells in that scenario.
Mm-hmm. Yeah, I wonder also, like, if anyone is interested in, sort of chipping in, how what are the next steps for cracking that 3D visualization problem? What tools, or what companies are best, situated to kind of approach that, to really get a 3D appreciation of what's happening in the islet, maybe even in vivo? I mean, is anyone close? Is there any kind of optical, innovation that's happening that could really drive that? Because I'm hearing it a lot, that it's an important understanding. I mean, it's important to kind of like go next level to really see that 3D if possible.
Well, I think it's possible. I'll give a shout out to, for example, Amelia Linneman, who's done phenomenal work imaging intact islets in situ. In a live animal. People have done this in the anterior chamber of the eye as well. There's benefits to that. It's impressive work, but whenever you take that in vivo, you're going to take a hit on your throughput. So there's always these trade-offs. What we can do is hundreds or thousands of cells, but clearly we're missing vascularization and innervation. So if you wanted to go faster and get 3D faster, you're looking at light sheet microscopes. That's possible. However, when you are approaching some of these platforms and say, well, great, I have my islets here and I want to now run my glucose media over it. They look at you like, okay, you want to do what now? You want to run buffers over these expensive microscopes. So what you'll find is that some of these setups are really made to image, but they're not always as conducive to doing the live imaging of the type that we do. That doesn't mean that can be resolved, but it's going to require standard setups that I think are available now, but they're going to require some retooling, maybe some parts making and some fabrication, and a microscope that you have access to, that you can tinker with, that you can basically build a perfusion system around if you wanted to do this kind of stuff at scale. But I'll also say the scale that we have now, we do hundreds of cells per trace and thousands of cells per day if we wanted to. So that throughput is already really quite good. If you wanted to do faster 3D, light sheet will do it, but then you're running into some of these practical limitations that I think are tractable, but we haven't in my lab. And I know Guy has himself unmuted. I don't know if you've been able to set this up in London or more recently in Montreal, Guy.
Yeah, so as you said, in the Nature Metabolism paper, we did that using a spinning disk microscope. We didn't go the full depth of the islet. We went 3 or 4 cells deep. And that was feasible with, we had enough photons to do that and still keep resolution and brightness. I strongly agree with you that the way forward is light sheet, and that is something that we are setting up and will attempt to do that in January with colleagues at McGill. So we see that as the way forward.
Fascinating.
Yeah, that's going to be very interesting. So I know that Gubra is all over the light sheet, so they are also interested in kind of that opportunity. I guess I would, I would ask like just one more sort of question about the gap junctions. How can you sort of further understand that functionality between, and whether or not the delta cells are connected by gap junctions, or if they're connecting to other cells via gap junctions? I mean, what can you just kind of drill down on that a little bit?
Sure. So I think the suggestion that delta cells are connected to beta cells via gap junctions of the kind that connect beta cells amongst each other, I don't think our data support that at all. And the experiments that I showed where we observe beta and delta cells at the same time, and then we come in and we basically prevent depolarization. And that characteristic calcium spiking that I showed, these delta cells always being on, the moment you prevent depolarization, that behavior goes away. And that means that a depolarization is enough to drive that behavior. And that depolarization would then propagate to neighboring cells that are connected to that cell through a connection 36 gap junction. It is the reason why beta cells are actually prevented from firing until they all do.
Yeah.
So in essence, the beta cell syncytium acts as a sink. So it does two things. What that does is it synchronizes the beta cells, but it also prevents them from going off until all of them do. It's almost like Dutch democracy, where nothing happens until you finally get consensus. And that's a slow mechanism where there is this inertia. And the fact that delta cells are able to behave independent of that, I think, is clear evidence that there isn't a gap junction. And if you drill down and look at the timing of it, the timing is off. Delta cells are not behaving at all in a way that I think is consistent with a gap junction connection. So I think there's papers out there on both ends of the equation, but I think our data, now that we have them in the same islets at the same time, I think are pretty clear that it's hard to reconcile with the connection 36 like gap junction mechanism.
Yeah, good. That was well said. I see you have a question here. Do you think if it is feasible to use GCAM P6 as a readout to perform a large scale screening for factors to improve calcium responsive stem cell derived beta cells?
Yeah, I think it is. Work I haven't showed. I mean, people are doing this. I know that Mike Sander and probably many others have cell lines that already have GCAM6 in it, so they can generate, they can differentiate them into beta cell likes and do those kinds of screens. I have yet to see approaches at scale, but I think in principle they could. We mostly on the mouse side routinely are doing a dissociated prep where we are imaging thousands of cells like in a couple of hours, just to get a sense of the functional heterogeneity. And that is actually in a way easier than doing an intact islet, because it's a 2D prep rather than a 3D prep. And that works well. Now we're not talking tens of thousands or hundreds of thousands of cells. We're still talking about thousands of cells, but thousands of cells in over the course of a day or two is actually, I think, a pretty reasonable throughput compared to what traditional techniques have been able to do. So if you had a targeted hypothesis or a limited number of conditions, I think this is eminently possible.
Okay. Last question for me is, what are your thoughts about layering artificial intelligence over some of these results that you've discovered and brought to light? Because it's almost like you're kind of, I mean, you look at some of this imagery and the readouts, it's almost like you've got an orchestra there of these cells, right? They're all doing sort of different things, but together they make this great contribution. So would artificial intelligence, I know there's companies like Immune AI in San Francisco and Tel Aviv, and then there's also Immune ML, which we just spoke to them the other day. That's an open source, machine learning platform. What kinds of experiments might be really interesting to do layering artificial intelligence over these kinds of data you have?
That's a great question, and we're starting to rely more and more on algorithms to kind of help us quantify like the differences in behaviors that we see. And I think the next step is, now that we have data and some data sets across thousands of alpha cells or thousands of beta and delta cells, one can and we look at these and we observe things, and then we go back in and we say, well, we think we observed this, and now let's isolate that particular aspect of the behavior. Let's put a number on that so we can quantify it and test whether it's real or not. The next step is, I think, to take all these data sets and kind of feed them in and say, okay, well, here are 5,000 random cells, and you go ahead and figure out how they resemble each other, group them together, and kind of tell us what different cells you recognize. And I predict if you do that within a whole islet, you're going to faithfully get delta, beta, and alpha cells back if you expose them to the right series of stimuli, like I showed you in these videos. But you can then also start to do within each, say, beta cell or each alpha cell population, you can then ask the question, okay, well, we know these are alpha cells, but is there anything that separates them within? And kind of let the algorithms tell you sometimes things that you might not have observed with the naked eye, and help you dig through these data sets that are getting increasingly richer. We haven't done this. We are still, we typically identify features ahead of time and then go in and quantify them. I think what you're describing or what you're suggesting is the next step and what we are positioned to do now.
Okay, then I'll connect you guys with the Immune ML team out of Oslo. They're very nice and
Great.
Really interested in collaboration. Okay, well, this was so fascinating, just amazing work you guys are doing up there at UC Davis, and just can't wait to hear what comes from the laboratory, the Huising laboratory next. Thanks again for joining us.
Thank you.